Welcome back to MLT 120. Today we're going to cover the introduction to granulocytes PowerPoint. These are white blood cells. We've been talking all about red blood cells so far, but we're going to start talking about white blood cells. Granulocytes include our segmented neutrophils, our basophils, and eosinophils. The first term we need to know and understand is leukopoiesis. This is the production of white blood cells. We've already learned erythropoiesis, which is the production of red blood cells, but now we're going to go through the whole process of the maturation and the production of white blood cells. Leukopoiesis. So where does leukopoiesis take place? Where are white blood cells normally produced? They're produced in the bone marrow primarily. The normal range for white blood cells in the peripheral blood, or how many white blood cells we usually have roaming around in our peripheral blood, is between 4,500 and 11,000 per microliter. So we don't have near as many white blood cells in our peripheral blood as red blood cells. The condition of having a decreased number of white blood cells or a decreased white blood cell count is called leukopenia. Having an increased white blood cell count is called leukocytosis. There are 5 types of normal white blood cells found in the peripheral blood, and these include our neutrophils, eosinophils, our basophils, our lymphocytes, and our monocytes, but only certain ones of these are considered granulocytic. These are our neutrophils, our eosinophils, and our basophils. And notice that they all end with "phil", meaning they have granules in the cytoplasm. So which ones are not granulocytic? These are our lymphocytes and our monocytes, and we'll talk about them a little later on in another PowerPoint. So just like we learned the erythrocytic maturation sequence, or the red blood maturation sequence, now we're going to have to learn the white blood cell maturation sequence. And remember, all the granulocytes in the peripheral blood are derived from the myeloid stem cell side in the maturation sequence, and we're going to look at that flow chart here that we looked at earlier on when we were talking about red blood cells again here in just a minute. But we can also refer to the maturation sequence of the granulocytic white blood cells as the myelocytic maturation sequence. So here's that chart or flow chart of the maturation of our blood cells that we looked at earlier on when we were talking about red blood cells. Again, we start out with our stem cell, our hematopoietic stem cell, and you divide out into the myeloid side and the lymphoid side. Today, for the granulocytes, we're going to be focused on this myeloid side. Later on, we're going to talk about the lymphoid side, and it's going to end up taking us down to the T cells and the B cells. But for today's lecture, we're worried about the myeloid side, because ultimately, we're going to end up down here talking about basophils, neutrophils, and eosinophils. These are your granulocytes. Okay. These 3 white blood cells right here. Remember, we also talked about this side, the myeloid side, with our erythrocytes, or our red blood cells. Okay, so we went from the hematopoietic stem cell to the myeloid. We've already covered the red blood cell. Now we're still on the side, and we're going to be talking about granulocytes, the white blood cells, the basophil, the segmented neutrophil, and the eosinophil. Okay, here are the stages in the maturation sequence for our granulocytes, or granulocytic maturation. You first have the myeloblast, then you have the promyelocyte, then you have the myelocyte, the metamyelocyte, the band, and then you have the mature cell, which can be a neutrophil, eosinophil, or the basophil. And again, this last step is where they're fully mature, and we're going to talk about each step in more detail on the slides that are coming up. But the good thing about this maturation sequence, there's only one nomenclature. So remember back when we were talking about red blood cell maturation and you had to learn 2 different naming systems, there's only one with this one, so lucky there. Before we get into all the different stages for the white blood cell maturation sequence, there's a few little things that you should know. All the granulocytes start as a myeloblast in the bone marrow. So the very first stage, you're not going to be able to differentiate between the different types of white blood cells, or granulocytes: the neutrophils, basophils, eosinophils. They're all going to look the same in this myeloblast stage, this first stage. And then they're going to progress on through the later stages, and then you're going to be able to tell what is turning into what type of cell. In the myelocytic stage, you're finally going to be able to kind of tell what type of granulocyte the cell is going to be, because in that stage the granules are going to have their own distinct appearance. Okay, so the neutrophils are going to have a certain type of look to their granules, same for the eosinophils, same for the basophils. So in the myelocytic stage, you're going to be able to finally start to differentiate. Again, they're still not fully mature, but you are going to be able to probably start telling them apart. And then lastly, the last bullet on this slide is the neutrophils are the most numerous in number for our granulocytes. So we're going to use our neutrophils as our examples for each stage of maturation in the next slides coming up. So each slide coming up is going to cover each stage of maturation for the granulocytes, but we're going to be using images of the neutrophils because they're the most numerous in our blood. The first stage in the maturation for our granulocytes that we're going to talk about is the myeloblast. And again, this stage you're not going to be able to tell one type of granulocyte from another in this stage. And I would encourage you to pull out your atlas and look at your atlas along with these slides. The myeloblast is on page 43, and you'll be able to see more images in your atlas than the PowerPoint or the video provides. So do look at your atlas as we go along at each of these stages, and it'll tell you which page to look at in your atlas on the slide. So for the myeloblast, and again, remember we're looking at neutrophils for our examples because they're most numerous. But the nucleus has a high nuclear to cytoplasm ratio. So remember that N to C ratio. So the nucleus is taking up most of the cell. Okay, you see that? There can be nuclei in the nucleus. The chromatin is fine. The cytoplasm has no granules, and the color is scant dark, and as far as dividing, mitosis can happen. The next stage is your promyelocyte. This is on page 45 in your atlas. Again, I would encourage you to look at your atlas so you can see more images. But in this stage, the nucleus is, again, you can see nuclei. The nucleus is said to be central, round to oval. So in the cytoplasm, we have what's called azurophilic granules. These help provide enzymes for digestion. These are those thicker, more dense granules you see, and they're usually by the Golgi. The Golgi usually doesn't stain very well, so if you can see a Golgi, it's usually lighter in color just because it hasn't stained. Also in the cytoplasm, we have primary nonspecific granules, and this is because we still cannot differentiate between whether we're going to end up with a neutrophil, basophil, or eosinophil. So those specific granules are going to start to form at the end of this stage, but for the most part we have primary granules and we can't differentiate between the three types of cells yet. Now mitosis can still happen, or the cell can still divide during this stage. The next stage is the myelocyte. This is on page 47 in your atlas. The nucleus is said to be off-center in the cell, and that is the case in this image we have here on the slide. You can no longer see nuclei in the nucleus. The chromatin is more condensed, so this chromatin inside the nucleus should be darker and more packed. The cytoplasm has secondary granules or specific granules. So the granules inside start to look more like granules that are in the cytoplasm of a neutrophil in this case, because we're looking at neutrophils. The cytoplasm can have a pinkish purplish color, and you have the myeloid spot. And the myeloid spot is the stage, or it shows up in the stage where it decides what it to be, but this is been visible throughout because this is the Golgi, and again, like I said before, it doesn't stain very well. As far as division of the cell, yes, this is the last stage in which the cell can still divide. The metamyelocyte is next. This is on page 49 in your atlas. The nucleus is starting to become smaller. The chromatin is even more condensed. There's an indentation in your nucleus that starts to form what is known as the kidney bean shape. Okay, so the nucleus is no longer rounded or oval shape, it's this little bean shape. The cytoplasm has specific granules in it, so we're going to be able to tell which type of cell it's probably going to be, whether it's going to be a neutrophil, basophil, or eosinophil. And the myeloid spot is usually still visible in this stage. The next stage before we're fully mature is known as the band stage. This is on page 51 in your atlas. The nucleus is usually more of an horseshoe shaped, or can even be an S shape depending on how the nucleus is kind of folded in your cell. The nuclear indentation is greater than 1/2 the width of the nucleus. The chromatin becomes more condensed, so you see how the nucleus inside is very dark purple, so it's very condensed, and then you have this U shape to the nucleus, or a little horseshoe shape. The cytoplasm has fine pink to tanned purple granules, and again, this is specific to the neutrophil because that's what we're looking at, or it's going to mature into a neutrophil. And you may see vacuoles in cells at this point. Now in the maturation sequence, we're to the fully mature cell, and we're looking at a fully mature segmented neutrophil, and this is on page 53 in your atlas. And for the neutrophils, the nucleus divides up into lobes. You can have 2 to 5 lobes or segments in your neutrophils. So this one has 3 segments. This one has 1, 2, 3, 4, and that one may be a 5th one. It's kind of hard to tell. But the segments are connected together through filaments, and the filaments may not be present, but this is what's called a little filament. This is connecting the different segments. The chromatin is very condensed in the nucleus. The cytoplasm, again, this is specific for a neutrophil, is going to be pink, tan, and purple granules. So it's going to be this very light color, and you may see vacuoles. A mature basophil is going to look different, and a mature eosinophil is going to look different than this mature neutrophil. And we'll look at those later on in another PowerPoint. Okay, so there's a couple more concepts we need to cover as far as white blood cells. We have something called a circulating pool and a migrating pool. The circulating pool is when white blood cells just float around and are in our peripheral blood. The migrating pool is when we have white blood cells that are adhered to vessel walls, but they can leave and enter the bloodstream. About 50% of neutrophils circulate, and about 50% are in the migrating pool. They kind of hang out in the vessel walls. And the reason that they do this is a term called diapedesis. This is when white blood cells leave the migrating pool and they travel into the tissues where they're needed. And this is in conditions where they need to fight off infection, things like that. Remember, the white blood cells are part of the immune system, and they help the body fight off and protect itself when needed. So So the white blood cells can basically leave that migrating pool when they're kind of called to action. Well, we have something called chemotaxis, and this is the attraction of bacteria and infections that stimulate that migrating white blood cell to leave the blood and travel to the tissues. So this is what calls those white blood cells to action. So kind of think of it as being at your county fair and you smell cotton candy and you're drawn to the smell to avoid the cotton candy is. And this is kind of what the chemotaxis does. It draws those white blood cells to where they need to go, to where the infection is, where they need to fight and help the body. So here's a question. So there's a 3-to-1 myeloid to erythroid ratio in bone marrow. So we have more white blood cells than erythroid in bone marrow. But in peripheral blood, we see millions of red blood cells in relation to just thousands of white blood cells in our peripheral blood. So why is this the case? This is because we need lots of red blood cells in the peripheral blood, because the red blood cells leave the bone marrow and reside only in the peripheral blood. And this is to help carry our oxygen to tissues and to take CO2 to the lungs. But the white blood cells leave the bone marrow, however, and do not all reside in that peripheral blood. Remember, we have those migrating pools. They most actually pass and live in the tissues and the organs, and migrate only where infections, or they're needed for infections or fight off infections. So this is why the bone marrow, we see more white blood cells in relation to the red blood cells. On this lecture for granulocyte maturation, we mainly focused on neutrophils because we mainly have neutrophils for white blood cells, but I would encourage you in your atlas to look at chapters 6 and 7 because those focus on eosinophils and basophils as far as what their maturation looks like. Again, those are granulocytes and it's the same steps, but you can see images of exactly what a eosinophil maturation looks like and a basophil maturation looks like, because we did not focus on those images in this PowerPoint and video lecture. So the eosinophil chapter 6 starts on page 56 and the basophil chapter 7 starts on 66. So I'd encourage you to look at those images so you see what a fully mature eosinophil looks like and a fully mature basophil looks like. So the maturation sequence steps are the same because they're granulocytes just like a neutrophil.